For the next presentation is mechanism of lipid droplet protein albumin targeting CRBP1 in pancreatic stellate cells to alleviate pancreatic fibrosis by Xinyi Yang from Naval Medical University, China.
Good morning, everyone. My name is Xinyi Yang from Naval Medical University in Shanghai. It's a great honor to share our recent work entitled Mechanism of Lipid Droplet Protein Albumin Targeting CRBP1 in Pancreatic Stellate Cells to Alleviate Pancreatic Fibrosis. Chronic pancreatitis is a progressive fibroinflammatory disease that can lead to long-term damage and even cancer. The activation of pancreatic stellate cells, PSC, is central to this process and is characterized by the loss of vitamin A containing lipid droplets. But exactly how PSC lose lipid droplets and become activated has remained unclear, and this question served as the starting point for our investigation. We knew that the loss of lipid droplets is accompanied by altered vitamin A metabolism. In particular, the key metabolite ATRA has been shown to inhibit PSC activation. This led us to ask, could specific lipid droplet-associated proteins play a role in regulating vitamin A metabolism and thus influence PSC activation? To answer this, we combined single-cell RNA sequencing and lipid droplet proteomics, which identified albumin, or ALB, as a promising candidate. We thereby hypothesized that ALB may interact with CRBP1, a key intracellular vitamin A transporter, to modulate vitamin A metabolism and ultimately suppress PSC activation and fibrosis. First, single-cell RNA sequencing revealed heterogeneity among PSC. A specific subpopulation, class 4, was enriched in lipid and vitamin A metabolic pathways and showed the highest expression of ALB, suggesting a potential role in maintaining lipid droplet stability. Indeed, ALB-positive PSC exhibit significantly higher droplet accumulation, supporting the idea that ALB may be functionally linked to lipid droplet preservation. Next, we performed proteomic analysis of lipid droplets isolated from both quiescent and activated primary PSC. It demonstrated that activated PSC had decreased ALB and increased autophagy pathway. Immunofluorescence further confirmed that ALB co-localized with lipid droplets, but was markedly reduced in CP pancreas. Taken together, these findings suggest that the loss of lipid droplet-associated ALB is a characteristic feature of PSC activation and pancreatic fibrosis. We also examined serum and pancreatic tissues from both CP patient and mouse models. Findings significantly reduced serum ATRA levels compared to controls. Consistently, pancreatic tissues from CP mice showed decreased expression of key vitamin A metabolic enzymes. These results collectively point to a disruption of retinoid metabolism during pancreatic fibrosis. To assess whether ALB is functionally involved in vitamin A metabolism, we used AAV8-mediated delivery to increase ALB expression, specifically in the pancreas. Remarkably, this intervention significantly attenuated pancreatic fibrosis, accompanied by reduced levels of collagen 1 and alpha SMA, two key markers of fibrogenesis. In parallel, we performed in vitro experiments using primary PSC. ALB knockdown promoted PSC activation and downregulated the expression of vitamin A metabolic enzymes. Conversely, ALB overexpression restored their expression and suppressed the activation. But does ALB's protective effect truly depend on vitamin metabolism. To test this, we employed inflammatory modulation. As expected, pancreatic fibrosis was aggravated in mice fed a vitamin A deficient diet, whereas it was alleviated by ATRA supplementation. Importantly, Alleviate ALB treatment reversed fibrosis in other conditions of vitamin deficiency, further demonstrating that ALB functionally links to vitamin A metabolic pathway. To understand how ALB works, we turn to molecular interaction studies. By performing CAR-IP silver staining, mass spectrometry, and spider assays, we consistently confirmed a direct physical interaction between ALB and CRBP1 in PSC, and notably, this binding was significantly reduced in activated PSC. Immunofluorescence also showed cytoplasmic colocalization of ALB and CRBP1. Moreover, ALB overexpression reduced autophagy-related lipid degradation, which means it may help protect the lipid store and slow down cell activation. In summary, our study identified albumin as a novel lipid droplet-associated protein that binds to to regulate vitamin A metabolism and suppress PSC activation. We are currently continuing to investigate how ALB is regulated and whether it could be developed into a potential therapeutic approach. This research is based on the chronic pancreatitis research team at Shanghai Hospital. Thank you for your kind attention.
Okay, we will continue the question and answer section.
Hello, welcome back. So, do you hear us clearly?
Yeah.
All right. So, in your experiment, you try to introduce your genes, right?
Yes.
Specifically at the pancreas, right?
Yes.
Okay. And then you showed the result that it's overexpression very well. Did you come across the expressions beyond in other tissues pancreas?
Yes, we do the single-cell RNA sequence and lipid droplet proteomics among and other tissues and organs. The results showed that the ALB remarkably upregulated in the pancreas organ.
Okay, all right, thank you.
I'm glad to answer.
Thank you for your excellent presentation.